Welcome to Nanocarriers Tryst with the lysosomes, implications in intracellular drug delivery and disease. I'm Anna, and it's my pleasure to introduce today's speaker. Dr. Gaurav Sahay is an assistant professor in the College of Pharmacy at Oregon State University, Oregon Health Science University in Portland, Oregon. Dr. Sahay completed his postdoctoral research with Professor Robert Langer and Professor Daniel Anderson at Koch Institute for Integrative Cancer Research at MIT. He received his PhD from the lab of Dr. Alexander Kabanov at the University of Nebraska Medical Center. Dr. Sahay's lab is interested in unlocking the molecular mechanisms involved in the transport of nanoparticles to deliver small molecules, nucleic acids, etc., inside cells. Now, before we begin, some basic housekeeping. If you have any technical issues, please use the question or chat box and we will do our best to troubleshoot. If you have any questions for our speakers, please use those same features and we will try to answer as many of them as possible at the end of the session. If we do not get to your question, we will answer them after the event via email. Now, without any further delay, Dr. Sahay, take it away.
Thank you so much, Anna, for the introduction. Today, it's my pleasure to talk about our interests and research in intracellular drug delivery. I would like to thank Precision NanoSystems for arranging this webinar, and it has been a very exciting time to work with Precision at many different forums. This talk today, I will basically focus on the nanocarriers tryst with the lysosomes and its implications in intracellular drug delivery and disease. So we all generally are working in the area of nucleic acid delivery. Some of us are also working in small molecules, etc. The nucleic acid field can be divided into DNA delivery, messenger RNA delivery, or small interfering RNA or messenger miRNA delivery. And basically, what my lab has currently focused on is using lipid-based nanoparticles for packaging these different nucleic acids so that they can be administered in vivo. Once they are administered in vivo, they hit different barriers. So one of the barriers which my lab is interested in is the intracellular barriers for drug delivery, namely the endocytic, endolysosomal compartments that pose a definite challenge for the release of these nucleic acid to the cytoplasm or into the nucleus where their targets lie. Lipid-based nanoparticles have been studied for a long time. What we are focused on are basically the systems that have reached clinical trials and have shown a lot of in vivo efficacy. These are composed of 4 different structures: a cationic lipid that binds to the negatively charged nucleic acid, a structural lipid, a PEG lipid for that presents stability, and then cholesterol also for stability, etc. So once a nanoparticle interacts with the cell membrane, it has different routes it can take to enter the cells. And these have been characterized to a greater degree into macropinocytosis, which is basically a non-specific mechanism, clathrin-mediated pits, caveolae, and clathrin and caveolin independent pathways. And the field of drug delivery has investigated for the past decade a lot into how the initial stages of internalization happen. It's at the later stage when once an early endosome is formed, the interaction of these nanoparticles with the internal milieu of the endosomes starts to form, and little is really known into how these interactions are happening, what changes are that bringing to the composition of a nanoparticle, and then how the internal environment of these endosomes and the architecture of endosomes affect gene or nucleic acid delivery. And as you can see here, that the early endosomes are much more tubular in structure. Most of the nanoparticles are then forced to the late endosomes, which are multivesicular in structure, and then they're degraded in a lysosome, which is multilamellar and has a low pH. Lysosomes have suddenly got a lot of traction into because they are not just a basic trash site, but people now have suggested that they might have much more bigger role than it was presumed. So my lab is interested in basically how these nanoparticles end up in the lysosomes, and then what kind of interactions are happening in the lysosomes. And perhaps that would answer our question of how can we deliver different therapeutics, especially for lysosomal storage disorders, which are rare genetic disorders. There are 50 kinds of them, and different dysfunctions in proteins lead to dysfunction in the lysosomal trafficking. So that's the interest and focus of my lab. Little is known of how these nucleic acid reach the lysosomal compartment and then escape. Some of the material escapes them. Is it during the process of trafficking, or after most of the material has reached the lysosomes? The field of drug delivery has suggested that with lipid nanoparticles, one way of exiting the endolysosomal system is by interaction of the positively charged lipid with the negatively charged endolysosomal system. That forms a hexagonal structure, destabilizes it, and that causes a lot of these materials, especially nucleic acid, to escape out of the endolysosomal compartments. The other theory which the field has also focused on is the proton sponge theory, wherein the pKa of a material lends itself to make the pH cause chloride ions to get thrown inside the endolysosomal system. It causes, because of the buffering capacity of these materials, that causes an osmotic pressure, and that leads to breakage of the endolysosomal system and causes rupture. This theory has been suggested for a long time, but little evidence suggests that there is some sort of a complete breakage of the endolysosomal system. The other theory is the umbrella hypothesis, which suggests that because of the changing pH, the polymer or a material can then disassemble, and that leads to the intracellular cytosolic delivery of DNA or nucleic acids. But having said that, most of these materials are trapped inside these compartments. This is just a very high multi-resolution imaging in which we tracked the transport of a lipid nanoparticles that was carrying an siRNA. And what you would see here at the end of this movie, and I'll not wait for it to complete, that the nanoparticle seems to keep on bouncing in and out of the vesicular structures. And because of that, it can sketch the whole vesicular structure. So at the end of this movie on your right, you'll see a big vesicular structure, which we think is a macropinosome. So this just suggests at a very deep nanoscale that how these materials are trapped inside these systems. New studies have suggested that siRNA delivery is also limited by endosomal escape. And the mechanisms of this escape has suddenly come to light, and more investigations though are needed, but we understand a little better of how siRNA escape. For example, Wittrup et al. In Nature Biotech showed that there is a very fast release of siRNA into the cell, and they were able to image that using a state-of-the-art microscope. What they were also able to find was that after the escape happens, because there is a slight disruption of the endolysosomal system, autophagy gets activated. And because of activated autophagy, then all the material is sort of further taken up and degraded in the lysosome. So it's not just the endolysosomal barrier, but autophagy also poses another barrier for escape from the cytosol. Gilleron and Marino-Zariel in Nature Biotech in 2013 suggested that there are two compartment two models by which an siRNA may escape to the cytosol. In one compartment, that the escape is happening from all different endolysosomal compartments as the trafficking proceeds. And in the other model, the escape is happening just from one system. So there is one vesicular system that is leaky enough for the siRNA to escape. They were able to further show this using in vivo and in vitro experiments that actually it's the second model that is true. In hepatocytes and HeLa cells, if you take lipid nanoparticles and have siRNA that is tagged with colloidal gold, they were able to, using electron microscopy, quantify the amount of gold. And that suggested that only 1% of siRNA gets escaped, and that leads to silencing. But there is a huge amount of siRNA that remains stuck in the endolysosomal compartment, and therefore this is a big challenge for drug delivery. Furthermore, our studies, when I was a postdoc with Professor Langer and Professor Anderson, suggested that these nanoparticles utilize the same transport mechanisms of recycling as does cholesterol, and a protein called Niemann Pick Type C-1 effluxes these. So on the right-hand side, you'll see a lot of accumulation of labeled lipid nanoparticles carrying siRNA in NPC1 knockout, but in the wild type, it's all effluxed out. And that, in the left, suggests that leads to much more silencing and much more translational activity. So this third study suggested the impact of recycling pathways in endosomal escape. My own lab currently has, when I moved to Oregon State University and Oregon Health Science in Portland, has moved into the area of messenger RNA therapeutics. And we were interested in understanding how the intracellular delivery of mRNA is happening. This field has exploded, and there are a lot of different people who are working in this area, a lot of companies that are working in this area that have reached clinical trials. And this can be used for cancer immunotherapy, infectious diseases, protein replacement therapy, and gene editing. So mRNA has become a new therapeutic modality that can be utilized for drug targets that were undruggable before. So we partnered with Dr. Shaolin Lan's lab, who has built his own 3D super resolution imaging systems that can image the internalization of messenger RNA. And what you see here is that labeled messenger RNA is on the surface of the cell membrane, and some of it reaches inside the cells. And we could quantify most of the mRNA inside some kind of a vesicular structure. You could even see some fusion of different vesicles happening at a very high res, and quantify how much mRNA is in the system. So we think endolysosomal compartment still poses a challenge for mRNA delivery. So to understand how the trafficking was happening, we started to utilize the haploid-based cell lines that are gene edited for different RAP proteins that are known for the biogenesis of early endosomes, of late endosomes, and of lysosomes, as well as of recycling endosomes. So what these cell lines can do is serve as a platform. So rather than having an assessment or on localization, we could quickly assess if formation of an early endosome versus lysosome was essential for mRNA delivery, not just for uptake, but functionally. So we then started to use these cell lines. And what we did was a very simple experiment in which we took lipid nanoparticles that were having a messenger RNA that codes for luciferase. We transfected these cells, wild type versus RAF4 knockouts that inhibits recycling, and looked for the functional activity of mRNA. That's the luciferase activity, and divided by the cell viability for normalization. And what you can see is a small decrease in activity in RAF4 knockout. In RAF5 knockout, which suggests that formation of an early endosome, we again see the red is the early endosome form, the wild type, and the blue is the RAF5 knockout cells. And you see a little bit of a decrease, but not significant. What was very exciting and strange, and we are still trying to figure out what's going on in this in this area, was when we utilized cell lines that were deficient of RAP7, that is involved in the late endosome lysosome formation, as well as it is involved in maturation of autophagosomes. That the wild type cells, you can see a very good gene expression, but in the knockout, it's completely obliterated. So we are currently investigating why is this case that late endosomes sort of, our formation of them, especially through RAP7 positive vesicles, is essential for mRNA delivery. And if you look at the electron microscopy of RAP7 knockouts, what we see is these enlarged endosomes. And what I think it's more of an early endosome because it's unilamellar, and the lysosomes are not forming in this case. So perhaps the formation of a lysosome is essential for some sort of delivery to mRNA. So we wanted to further test whether this was something endocytic transport related or something else that RAP7 was doing. And to bypass the endolysosomal system, we electroporated the mRNA in the knockout and the wild type, and then used different pulse voltages, width, pulse numbers. And what you can see again is that even bypassing the endolysosomal system by using a RAP7 knockout cell line and by electroporating, you find the same sort of obliteration. So right now we are still figuring out what kind of downstream signaling is happening that is essential from a lysosome for gene delivery. So finally, based on the mechanisms of intracellular delivery, we think that the biogenesis of the lysosomal compartment is very essential for mRNA delivery and transfection, and we are trying to figure out why that is and what's going on in that area. We are also working with different genetically edited cells, and we think they offer a quick platform to assess different trafficking pathways for multiple platforms, not just one, and very quickly at a functional level. The next part of the story, we wanted to understand if we can improve the intracellular delivery of by enhancing endosomal escape. And the vesicular compartment usually is formed of proteins, but also of different bioactive lipids. So we thought if we took a bioactive lipid library and thought of using that as a precursor for enhancing the permeability of these endolysosomal system, we'll see a better effect. So graduate student in my lab, Siddharth, carried out the earlier studies and this study as well, where he used around 212 bioactive lipids and the same luciferase expression assay, and saw different changes in the activity once the LNPs were co-delivered with these bioactive lipids. So we found different drugs, and because of patenting reasons, I just say drug X here. We took different amount of drug X and then transfected lipid nanoparticles. We see an increase in gene delivery. 2 out of 3 analogs of drug X also showed a better transfection efficiency. What I like about bioactive lipids is that you can formulate it with LNPs very easily as compared to other molecules that might have difficulty with it because it's a lipid-based platform. And so we were able to quantify if there were any changes in the physicochemical properties in terms of the size, and we don't find much change in terms of the encapsulation efficiency, and we are pretty good in encapsulating with or without the drug. Or in the presence of Triton to break apart these particles, we see a very similar sort of an effect with both the particles and particle with the drug. So we are able to formulate these bioactive lipids with the RNA, and once we now have them in the formulation with the LNP, the earlier one was just a mixture, we start to see increase in HeLa cells as well as HepG2s, around 2 to 5 fold. And then using the same formulation, then we compared if we can improve the mRNA delivery in vivo. LNPs have a certain kind of affinity to the liver, and you start to see gene expression within cells of 0.1 mg. And in the presence of drug X, also we see around twofold improvement in the gene delivery. So at this point, it's a start. We hope to find other materials, other bioactive lipids that can improve further the intracellular delivery of messenger RNA. So to hold this part, I'll say that the endosomal escape pathways are still under investigation. There are multiple ways by which intracellular delivery can happen with bacteria and viruses, and might be true with nanoparticles as well. This may include some active transport from the vesicles, some lysis, back fusion of the multivesicular bodies to the cytosol, and as well as direct fusion from the cell membrane. So this part of the talk was related to intracellular delivery of nucleic acids. But based on our findings from my previous research, we knew that nanocarriers are very effective, therefore, in delivery to cells that are deficient of NPC1, which is basically a lysosomal storage disorder. So NPC1 is a protein, a 13 transmembrane protein that sits on the late endosomes, lysosomes, and is the cholesterol that enters from the extracellular milieu ends up in the lysosome and is recycled out or effluxed out by NPC1. Absence of NPC1 leads to a huge retention of nucleic acids. So based on that theory, we thought, would nanoparticles be effective carriers for treatment of Niemann Pick Type C-1 disorder? So absence of Niemann Pick causes a huge accumulation of cholesterol in the cells. And because of the amount of cholesterol that gets accumulated intracellularly, the most affected parts are the brain, that's the cerebellum, in which there is a loss of Purkinje fibers. So if you look at the wild type's brain of a mice model, you see these Purkinje grape-like structures. Absence of NPC1, they're completely gone. The same happens with hepatocytes, and you see a traffic jam sort of a situation because of absence of this late endosomal protein, and a high amount of cholesterol causes this degeneration. And if you look at the mice, they have ataxia and they die within like 2 or 3 months. The issue with this disorder also is that it affects children. So basically a child that has deficiency of NPC1, they start to develop neurodegeneration. It's called Alzheimer's for kids. So you see a slow sort of lapse of memory, ataxia, cognitive functions, and death. And the median age of survival for these kids is 20. So there's no drug available for treatment of this devastating disorder. One drug which has reached clinical trials is cyclodextrin. And this is an untreated cat that has absence of Niemann Pick Type C-1 protein. It also has the same ataxia and shaky features. People in the Niemann Pick field have figured out that a small molecule called cyclodextrin, which in the pharmaceutical industry has been used as an excipient for solubilizing different drugs, by itself has tremendous effect. So if cats are given intrathecal injection of this drug, that leads to the cat to live for more than 2 or 3 years. In case of the wild type cats, they die within months. So this is a huge sort of a breakthrough, and NIH started a clinical trial on it, which has reached phase 2. The challenge has been the amount of cyclodextrin that has been given is around like 900 milligrams in kids. and it's a biweekly injection, and it doesn't cross the blood-brain barrier, so it's given intrathecally. So we thought, can we use first nanotechnology, because nanoparticles have shown this tropism for the lysosomes to deliver cyclodextrin so that cholesterol can be solubilized out. And can we then in the future use these carriers to transport across the blood-brain barrier so that you can just give this drug IV. So this work was done by my postdoc who has joined me here, Dr. Anna Brown. And initially, we just utilized the NPC1 knockout cells, and the red stain is Philippin that stains cholesterol that is accumulated inside the cell. So wild type cells have no cholesterol, blue is the nucleus. Knockout, you start to see a very large amount of accumulation of the cholesterol inside the cells. When you transfect this with RAB7, an early late endosomal marker, we start to see for a big, large vesicles and the red cholesterol or Philippin stain inside it. So for sure, the cholesterol is sequestered inside these enlarged late endosomes, lysosomes, and is not effluxed out because of the absence of Niemann Pick. In the wild type cells, you don't see these large structures or the accumulation of cholesterol in just these vesicles. So we started to utilize different methods. The first thing which we did was we utilized cyclodextrin and saw that cyclodextrin by itself can clean out cholesterol from the cells. So there was a lot of the studies which were done before were exactly right, that utilizing cyclodextrin on its own does show clearance. The next part of what we started to do was we started to make liposomes which had DSPE PEG in them, and it also had HSPC and other lipids, and we started to formulate these 2-hydroxycyclodextrin in these liposomes. We tried to play around and increase the concentration of DSPE PEG in these formulations, and through serendipity, Anna found that DSPE PEG on its own does clearance of cyclodextrin from the cells. So we thought of using liposomes as carriers, but we found DSPE PEG, which has been used as an excipient, is already a part of FDA-approved drug nanoparticle called Doxil, and it was a surprise to us that it can clear out cholesterol on its own. So if you look at these structures, they are around 12 nanometer micelles. Then addition of different concentrations of DSP PEG causes a flux of cholesterol, and you start to see a decrease in the level of Philippin staining inside the endolysosomal system, and you can quantify the whole amount of cholesterol inside the cells as well, and you see a very similar trend. So we are able to sequester out cholesterol using just DSP PEG. And to our surprise, if we take a labeled DSP PEG, throw it on cells, and then fix the cells at 2, 3, 4, and 6 or 24 hours, we see enhanced accumulation. So the data here, which has not been shown, is of wild type cells. You don't see any accumulation or it's effluxed out. But in knockout cells, just labeled DSP PEG, you start to see a large accumulation. Then we stained it for Philippin, which marks the late endosomes, lysosomes, as well as cholesterol in Niemann Pick Type C-1, and you start to see a huge co-localization of DSP PEG with the lysosomal compartment. And we think because of this direct interaction of DSP PEG in the lysosomes, that might cause solubilization or formation of some sort of liposome within these structures that causes efflux of cholesterol. And then if we simply then mix cholesterol with cyclodextrin, cyclodextrin alone at higher concentrations cleans out cholesterol. But if we just mix it with DSP PEG, not form a particle, nothing like that, we start to see a synergy that happens. And this was done using a high content screening system. We took like 48 images n equal to 3, and were surprised at how close the data was and how well the synergy is. So later on, we started to use different peg lengths, different amount of liposomes and things like that, and it is clear to us that just DSP peg can serve as a very good sort of an excipient in a cocktail of these different drugs for NPC1. And this paper has now been accepted in scientific reports. Our first paper as corresponding author, and Anna's first paper from my lab. And so we are very excited to take this work in vivo, where we hope to perhaps directly inject these components to the brain, and then also develop new ways of delivery across the brain. So the current questions regarding bioactive nanocarriers is that if different lysosomal storage disorders based on the tropism of these nanocarriers can be utilized for delivery of small molecules, genes, as well as diagnostic and imaging tools. We have partnered with the Niemann Pick field, as well as Professor Newald from OHSU, to come up with ways, because most of these disorders are neurodegenerative, so that we can cross the blood-brain barrier also in the future. Our hope is that just with the nanocarrier, the distribution within the brain, even if it's directly injected to the brain, is much higher, as has been shown by others in the field of drug delivery, than the drug alone. So the lab basically now has, the main focus is intracellular delivery. We are interested in basically understanding the endosomal escape pathways, but also we are interested in developing new probes to understand the microenvironment of lysosomes, and we are interested in delivery of treatment and develop therapeutics for lysosomal storage disorders. Finally, I'd like to thank my lab, which has just started to grow and develop and buzz. I'm especially thankful for Dr. Anna Brown and her work on lysosomal storage disorder, and Sid Patel, who has done most of the work for intracellular trafficking and the bioactive lipid parts. And I'm also thankful to the other members, past and present, of my lab. We are collaborating with other different labs to go into this direction, and we thank for the support by the OSU College of Pharmacy, Moderna Therapeutics for the mRNA work, the AACEP, and the OSU Foundation. And I hope to have questions from you, and thank you so much.
Thank you, Dr. Sahay. Now we have time for a couple of questions from the audience. Can you comment on the impact of endosomal escape versus RNA release rate from lipid nanoparticles? Namely, does RNA release from nanoparticles represent a limiting step?
That's a great question. I think that is definitely a limiting step. Based on our experience, we have, and this was during my postdoc time, me and another postdoc, Chris Alabi, at that time, who also now is a professor with Dan and Bob, were trying to figure out this question, and we used FRET-based probes, and it was clear that a nanoparticle shouldn't hold the cargo too tight, but also not too loose that it just breaks off. But having said that, what is happening inside the endocytic environment, the rapidly changing endocytic environment, is unknown. And I think super-res imaging and other imaging platforms, which we have at OHSU and the Collaborative Life Science Building, might offer some tools that we can answer this question. But I think both are very essential, and we don't really know how the disassembly is happening, and whether there are other components reforming within this environment. So that's a big question which we still have to find an answer to.
You showed that mRNA lipid nanoparticles accumulate in the cell membrane. Do you have an explanation for this?
So the answer is no. The interesting part is that the same formulation with the lipid nanoparticles, if you use for siRNA delivery, you don't find that huge accumulation on the cell membrane. But if you use mRNA, we have seen that. The picture I showed you was actually of a human NPC1 knockout cell line. So there the accumulation is even higher. But it is still unclear to us why the cargo and changing the cargo, maybe the shape changes, is affecting how their intracellular distribution happens, or maybe different proteins are binding when the cargo is siRNA versus mRNA, and their trafficking pathways are different as well. So that could be another answer to that.
Thank you. Thank you so much for joining us today. If we didn't get to your questions, we will answer them via email after the event. If you have any questions or would like to learn more about the NanoAssembler, visit www.precisionnanosystems.com. And don't forget to follow us on LinkedIn and Twitter. From Dr. Sahay and myself, have a wonderful rest of your day, and thank you for your time. Cheers.